Dr. James A. Shapiro from the University of Chicago. He's a professor of microbiology emeritus in the Department of Biochemistry and Molecular Biology. He received his PhD in genetics from Cambridge University in 1968 under Professor W. Hayes, FRS, at the University of Chicago. Since 1973, he was the Darwin Prize Visiting Professor at the University of Edinburgh in 1993. In 2001, he received an OBE for services to the Marshall Scholarship Program. He is the founder of www.thethirdwayofevolution.com, intended to raise awareness of scientific alternatives to intelligent design and neo-Darwinism. His pioneering books are on mobile genetic elements, natural genetic engineering, bacterial multicellularity. His complete CV can be found at https shapiro.bsd.uchicago.edu/cv.shtml. Dr. Shapiro is also a direct student of Nobel laureate Barbara McClintock and the amazing work that has been done there. And we're very grateful he could join us today.
Thank you. Can everybody see that?
Yes, we can.
And you can hear me?
Yes.
Yeah, so I changed my subtitle since I sent it in to you, so it would say more explicitly what I meant. And what I'm saying is why evolution works. Life doesn't wait for accidents to happen. Life changes itself. So it changes the biological process. And I want to go through some of the evidence for that and what we've learned about evolutionary change over the past several decades. Evolution is descent with modification, which is a correct description of it. But we have to ask, how does cellular and genome modification occur in evolution? And I think that the random mutation natural selection scheme doesn't really work. It happens organically, biologically, not randomly. There was no space for biology in the classic neo-Darwinian formulation. And if you just think about the numbers, random mutation is not an adequate explanation for evolutionary variation. There are very vanishingly small probabilities of generating significant code. If we assume an absurdly high 1% mutation rate, 1% positive mutations per generation, by the time you've made a sequence of 10 base pairs, your probability has gone down to 1 in 10 to the 20th. That's 1 in 10 million, billion generations, which is impossibly small. So random changes are not going to work. It has to be some more organized process. And as you'll see in a minute, 99.9% of incorporation errors in the DNA are removed by replication proofreading functions. So errors do not make important variations. And more than 99% of so-called spontaneous mutations are actually enzymatically induced by mutated polymerases, cytidine deaminases, and other DNA modifying enzymes. So they're actually of biological origin. Now, this slide illustrates how the replication proofreading occurs. Here's a template strand of DNA with its bases C, G, A, T, and so forth. And here's the new strand being synthesized where these complementary bases are being attached. And these first 6 here are all correct. But now the polymerase is going to add a T across from a G, which would be incorrect. and that distorts the structure of the double helix. And the polymerases, which do DNA replication, are sensitive to the structure of the helix. And when they sense a distortion, they change their activity from being a polymerase to being an exonuclease and chew back and remove the incorrect nucleotides. And then they recover the ability to proceed with polymerization. So this removes 99.9% of the, at least, of the misincorporations. And after replication is complete, if there's still some misincorporated nucleotides, like here, there's a process called post-replication mismatch repair, which senses the distortion in the helix at this site, and the protein which senses that calls in other proteins which remove that region and remove a section of polynucleotide, and that is then replaced by DNA polymerase correctly, and then it can be sealed up by a DNA ligase. So spontaneous changes and replication errors are kept to a very low level. And as I said, it's things like mutated polymerases, which introduce the mutations. If you remove those genetically from an E. coli strain, its spontaneous mutation rate goes down at least 2 orders of magnitude. Now, there are many events in evolutionary change which require cleavage and ligation of extended DNA segments. That's where DNA is cut and spliced, just as we do in our genetic engineering efforts. And that's why I call these processes natural genetic engineering. Genetic engineering, which is built into all living cells, and the systems they have for doing that vary from cell to cell, but they all can do it. And there are certain results in evolution which we know have required the cleavage and ligation of extended DNA segments. That is, DNA segments longer than just a base pair or two. And there are horizontal DNA transfers. These have been found in doing phylogenies of genome sequences, where it's found that all of a sudden in a lineage, a new activity appears. An example is in nematode worms and beetles, which have been adapted to digest plant material. And plant material is very difficult to digest because it has complex polysaccharide polymers. And bacteria and fungi have these enzymes. And it's seen that nematode worms and beetles acquire the sequence for one of these enzymes all together at once in a single-step acquisition from these microbial sources. And these kinds of transfers occur across all taxonomic boundaries in all directions. And for this to occur, the DNA has to be excised from the donor cell, from the donor genome. It has to be transferred it has to be excised, it has to be transferred to the recipient cell, it has to be acquired and then integrated into the recipient. And that requires a great deal of natural genetic engineering to occur. And this is very, very, very common. And the more we learn about genome sequences, the more frequent we find horizontal transfers to have been. There's another part that I'll show you some figures to illustrate, are the rearrangement of protein domains. Proteins are not unitary molecules. Comprised of sections called domains, which have their own structure and their own set of activities and interactions. And they can be found, the same domain can be found in different proteins, and they can be rearranged. And this gives a kind of combinatorial functional process of change by domain acquisition or loss, which is much faster than single nucleotide modifications to change the activity of proteins. And domain acquisitions and rearrangements can occur after viral endogenization. New coding sequences are brought into the genome because virus sequences are quite different from cellular sequences. That's a mystery which we don't yet understand, where the virus sequences originated. It also carries transcription signals and other expression signals. This is seen in, for example, in endogenous retroviruses, playing a role repeatedly in mammalian evolution and placental development. And also there's rewiring of transcriptional networks by transposable elements, and we'll see some more details on this. Transposable elements were discovered by McClintock in the 1940s, and they're stretches of DNA which are capable of moving from one place in the genome to another. And there are a bewildering array of these elements that have been found. I'll show some of them in a few minutes. And what they can do is they can carry with them control sequences and insert them at different locations in the genome. And if two or more of those elements insert near coding regions, then those two coding regions can respond to the same control sequences and to the same transcription factors. And it's a way of conferring coordinate regulation on multiple sites in the genome and building up regulatory networks, which are needed for development, because most development is network development, as we just heard, rather than simple gene changes. Finally, there are chromosome rearrangements which occur, and it's the formation of double-strand breaks in the chromosomes, and those are repaired by end-to-end joining of broken ends. And there are alternative repair systems for doing that, and I'll discuss those in some detail, how they work. Some of the results have been quite astonishing in the last decade. It's been very, very interesting. So here's a figure from the initial DNA sequencing of the human genome in 2001, figure of different growth of domains in proteins which bind to chromatin in different ways. And you see here, for example, in A, does my cursor show up on the screen?
Yes, it does. We can see your cursor.
Okay, good. Well, if I can control it properly, it will be helpful. So this is a protein from yeast, which is a chromatin protein. By the time it's gotten into worms, flies, and vertebrates, this domain, this bromo domain, has multiplied sixfold, and it's acquired other additional domains, an HMG domain and these two zinc finger domains here. And so it's a much more complicated protein, which has a much more complicated set of interactions and control responses. Here is the protein which is found in yeast, flies, and vertebrates. In yeast, it has this basic structure with two PhD domains. I'm not sure what those are. And these two EP domains on the ends. In worms, it acquires a bromodomain. In flies and humans, it acquires zinc finger domains and a BMB domain. And finally, in worms, you can find an even more complicated version, which has a totally different domain structure at the end. And here's a common ancestral protein, chromatin binding protein, which in humans acquires these three triangular domains, this diamond-shaped domain, and has the bromodomain and the PhD domain, as well as the set domain at the end here, and these extra copies of this domain and the thread domain. So that the proteins change in groups of extended regions, sometimes hundreds of amino acids. And here is another figure from the same paper, which is the expansion of domains and architectures of transcription factors. And down here, we just see that there are transcription factor domains, 2 and 3 and 4 domain factors, which are shared by all organisms. And here we see that flies and humans share these architectures of different domains. And the fly has a special thing, which is multiple copies of this A domain. And worms has a double copy of this, I think it's EDF domain. And here are domains that are unique to human beings. So protein structure and protein evolution is completely changed in its our understanding of how it occurs. Now, here we come to transposable elements. And those are elements which can move from one place to another in the genome. Barbara McClintock called them controlling elements because she found that they changed the regulatory properties of regions of the chromosome where they inserted. And she was correct, as you'll see in the next slide. But here, they're just showing the basic architecture of the different kinds of transposable elements. And what you see here are DNA transposons. And there's, as I say, a dizzying array of DNA transposons, which basically have two characteristics: inverted repeats at the ends, which is where the DNA is cleaved, and a transposase protein, which does the cleaving of the ends and the ligation to the target sequences. And this happens in such a way that a short segment of the target sequence is duplicated. And this is called the target site duplication. And that's indicated by these red ovals here. There are also transposable elements which move about through RNA intermediates. And there are 4 variants of the they're called retrotransposons, because they move by making RNA and then copying it by reverse transcription into DNA. And one of the most common types are retroviruses, which have what are called long terminal repeats at the ends. It allows them to insert into DNA, and they have a complex overlapping protein coding structure, which allows them to make reverse transcriptase and endonuclease activities for integration into the genome and for converting RNA to DNA. And they also contain an envelope protein, because all this takes place inside a virus-like capsid, which has a membrane envelope around it, and which contains the envelope protein. We'll see why this is important later in evolution, because it plays a role in an important role in mammalian evolution. The red sequences are the target site duplications when the LTR retrotransposons insert. There are also what are called line elements, long interspersed nucleotide elements, which don't have the elaborate structure of the LTR retrotransposon. They just have two ORFs, or open reading frames, encoding an endonuclease activity and a reverse transcriptase activity, and an ORF1 protein, which we don't yet know what it does. And that can be transcribed and inserted virtually anywhere in the genome. And the process is sequence independent, both for the target site, except that it has to have a run of T's to pair with the poly A's at the end, at the 3' ends of the molecule. This is important because the same enzymes can take any messenger RNA, which has poly A at the end, and insert a reverse transcribed copy into the genome. So all of the coding regions of the genome are in principle transposable. And again, they make a target site duplication. And finally, here at the right, I can't see it, but because the figures, the individual images are covering it, but there are sign elements, which instead of being long interspersed nucleotide elements, are short interspersed nucleotide elements.
The numbers read 5664 1626 1632.
What number?
You're trying to point to the number on the bottom left-hand screen?
No.
Okay, I apologize.
Those are just the page numbers, I think, for the article. I was trying to point to the sign element, or the ALU element, at the lower right of the figure. There we go. And these lack the coding capacity of the line elements, but they're able to be transcribed by those proteins and to move from one place to another. And they're often the most abundant components of the genome, as they are in ours. And here's another view of the transposable elements in the human genome. This is from the same initial sequencing and analysis of the human genome in 2001. The lines are present in about 850,000 copies. That comprises 21% of the genome. The protein coding capacity of the genome occupies 1.5% of the genome. So there's much more of this DNA than there is of coding DNA. The same is true for signs. There are 1.5 million of those, although they're much smaller. And they comprise 13% of the DNA. And then the retroviruses, like the retroviruses and the non-autonomous derivatives of those, is about 450,000 for 8% of the genome. And the DNA transposons, which are fossils, are not active anymore, are 300,000 in number for about 3% of the genome. And for a long time, this was thought to be so-called junk DNA or selfish DNA and not to be functional. But we now know that all of the genomes of mammals and other complex organisms are transcribed in cell-specific ways, and they encode thousands and thousands of non-coding RNA molecules. which carry out all kinds of regulatory and important functions within the cell. And we've entered the period of RNA molecular biology. And there's a very good book out that can be downloaded from the internet by John Maddock and Pedro Amaral on that subject. And that's going to change the way people think about genomes and what they do. We don't yet understand how all these repeats can be part of these functionally different molecules, but the fact is that they are. Now, here I want to show you one of the poster children for conventional evolutionary theory, the spotted butterfly, British peppered moth. Shown on the left is the original wild type. And in the industrial age, in the Industrial Revolution, trees and buildings were covered in black soot. And so it would be easy for predators to identify these peppered moths. So they became all black. And this was thought to be an example of natural selection at work. But it turns out when the molecular biology of this was studied, that the region giving the black phenotype was formed by the insertion of a DNA transposon into a locus called cortex, which is involved in producing the surface of the moth. And for some reason, it becomes totally covered in melanin. And what you can see at the ends here in red are the ends which have their termini, which have their inverted repeats. And then here, just adjacent to those ends, is the target site duplication, which is characteristic of the way these transposons move. So I think it's ironic that what was supposed to be the proof of the natural selection idea turns out to be a transposable element insertion. Now here's a figure from a paper by Nishihari, who's one of the specialists on the function of these elements in producing functional networks. And here is a picture of how an enhancer region is built up in the mammalian lineage. The ancestor of mammals has a sign element at one location. And at the next stage, in a Therian ancestor, I don't know what Therian means, but it's an older form of mammal. It picks up a block of DNA next to it. And then in the Eutherian ancestor, that's the ancestor of all mammals like us, which have normal pregnancies. Another second transposable element inserts nearby, and that creates a regulatory region which directs expression to the nasofrontal area of the mammalian embryo. So that's one of the ways that these transposable elements can build up control sequences. There's a sign element in some mammals called the B2 sign, which contains binding sites for the CTCF transcription factor, which is also involved in forming cohesion bonds around adjacent chromosome regions. And that spatial proximity is very important, both in cell division and in genome expression, because there's often interactions between regions, enhancer regions and transcription regions on different proteins. Here's an example of a retrotransposable element, MER41B. MER means mammalian endogenous repeat, and it carries enhancers for various loci and inserts them next to them and provides the same control. It's coordinated control that I mentioned earlier. And finally, there's one of these line elements, which has picked up binding sites for 4 different transcription factors, and it binds next to different loci and allows them to interact with all of those different transcription factors. And this has been important during the evolution of mammary glands. Again, a live birth function. And it's an interesting case of polyvalent regulation going on. Oh, I'm going the wrong way. Now, I want to change subject a bit and talk about chromosome restructuring. This is based upon the discovery made originally in 2011, where it was found that cancer cells and then germline cells could generate novel genome sequences, novel chromosome structures at multiple sites all at the same time in one cell division. And some of this has been verified by direct examination in cell culture experiments. Some of it we know only from genome sequences. But chromosome restructuring requires the joining of 2 broken ends of 2 different chromosomes, or the broken ends at different positions of the same chromosome. And every chromosome break, repair, which joins those ends together, generates at least 1 chimeric genome sequence. Now, what's the importance of a chimeric sequence? It is that it can produce novel molecular encoded products. For example, having one end of the protein molecule be from one genetic locus and the other end of the protein molecule being from the other, and it would scramble the domains in the 2 proteins and create a novel sequence, and or altered patterns of regulation of genome expression. Because as we've seen, genome expression is controlled by factors which recognize sequences near the transcribed region. And as we'll see, alternative end joining, as the mechanism that joins the broken ends together, can generate multiple novel chimeric sequences by DNA polymerase template switching during the repair process. This is so bizarre that if anybody had dared suggest this 10 years ago, they would have been laughed at as proposing something that's preposterous. But it actually happens, and it's well documented now. Chromosome restructuring can alter the 3-dimensional arrangement of multiple chromosomes in the nucleus and interrupt or alter a programmed sequence of genome expression. And what I mean there is, because chromosomes can be near each other and affect the expression of sequences from another protein that's nearby, if you change the 3-dimensional arrangement, you change these regulatory interactions, and the result is you get a different program of gene expression, and you certainly will have a phenotype, and possibly one that is useful for a new evolutionary invention. And finally, chromosome restructuring is important because restructured chromosomes don't pair the same way as their parents did. And if they're half of a diploid cell, they interfere with meiotic pairing and create a barrier to gamete formation and sexual reproduction. And sexual reproduction is important because one of the definitions of a species, or two different species, is that they're reproductively isolated from each other. And so chromosome structure matters very much in reproduction of higher organisms. Now, what you're seeing here is what's called a circus plot, a circle plot, and it shows the human genome around the outside of the circle with the chromosomes in order, chromosome 1, chromosome 2, chromosome 3, and they're in order of size down to chromosome 24. And then there's the X chromosome and the Y chromosome. And these lines indicate where there have been breaks and rearrangements in the chromosomes. And it's been found that you get these rearrangements that come in multiples. And what these 4 slides illustrate is what's called chromothripsis. And that means chromosome shattering. And what this first slide shows is a colorectal cancer cell with 239 rearrangements of chromosome 15, and well, it has other rearrangements as well, some of which are interchromosomal. Here's a thyroid cancer cell with 77 rearrangements on the short arm of chromosome 9. Here's a renal cancer cell with 55 arrangements to chromosome 5. And here's a lung cancer cell line with 85 chromosome rearrangements on chromosome 8. So these are chromothriptic arrangements which have a limited region, but they create complete rearrangement of all of the DNA in the chromosome. You'll see this happening in a video in just a second. A completely different pattern was found and described in this first paper from 2011, is where you have a limited subset of the genome. In this first figure, there are 5 chromosomes, and they have multiple interactions and restructuring across chromosomes, as well as within chromosomes. And the rearrangements are not randomly distributed, they're clustered in certain regions of those chromosomes. And this fits with how we know these chromosomes behave during the cell cycle. They come near each other and form what are called topologically associated domains. And here are some more examples with 3 different chromosomes. This one has 4 different chromosomes. So this is a very different kind of rearrangement, and mechanistically, it's going to be very different from the chromothripsis. And we don't understand it as well as we do chromothripsis. Let me get back to where I should be. And here's what this looks like in a video recording of a cell culture experiment. Here is the nucleus of a chromosome of a cell, and it's mostly colored green. The chromosomes are undergoing DNA replication. And this small red structure here is a micronucleus containing a fragment of one of those chromosomes generated by something like CRISPR cleavage. And as you'll see, as the cell division proceeds, the chromosomes will condense and pair and separate in the main nucleus. And you'll see what happens in the micronucleus is that the chromosomes shatter and then a period undergo some form of replication because they turn bright green. I hope you all saw that. It's very dramatic. I'll do it one more time about how the micronucleus all of a sudden fragments into dozens and dozens of small pieces. And that's how chromosynthesis occurs. Now, as people have studied these multisite chromosome rearrangements, they've characterized some of the processes that occur. And what I'll show you is what's called template switching, which occurs when DNA rearrangements occur by a replicative process involving a DNA polymerase, which joins together broken ends which have been paired down on single strands. And here was a chromosome 6 from apparently from the parent or a progenitor of a patient with a neurological disease that requires the presence of these regions crossed out next on chromosome 6. And here's the broken fragment of chromosome 6. And the polymerase first joins it to this region of the X chromosome. And it acquires this sequence next to chromosome 6. And then it jumps back to chromosome 6. And then it jumps to this region A of chromosome 6. And then finally, it terminates by joining to an end fragment of chromosome 6, which has a telomere. And the result is a chromosome which is altered significantly in structure, has duplicated copies of region A, and has an extra copy of region B than the one on the X chromosome. So this is just a simple example of what's called template switching by these polymerase-dependent end joining processes. And they're quite mind-boggling when you think about it because they create chimeras within chimeras, and they have a virtually unlimited capacity for generating new sequence. In fact, in some cases, the polymerase makes sequences which have no homology with any of the parental genome. And it's concluded that, and it's been demonstrated, that the polymerase has put together untemplated DNA by terminal transferase activity. And we know that one of the polymerases involved in this process has robust terminal transferase activity. So not only do they join sequences together in ways that they weren't ever joined before, but it can add completely novel sequence. Now, what relationship do these changes have to speciation? And it turns out we know how to make new species and make them quickly. And that's called hybrid speciation. G. Ledyard Stebbins, one of the founders of the modern synthesis, wrote a 1951 article about it. It's called Cataclysmic Evolution. He thought he was writing about an exceptional situation, but as we were learning over time, this is more and more common among living organisms, animals, plants, yeasts, all together. And what hybrid speciation means is when two species which are related but different or distinct mate with each other, the genome of the germline of the resulting progeny is unstable and undergoes rapid changes in chromosome structure, activation of transposable elements, and can, if properly bred, generate a new species which is reproductively isolated from the parent species within 2 or 3 generations. And this occurs in these heliconius butterflies, as illustrated here in this picture, showing related mimicry patterns between the butterflies. And it occurs in a wide variety of different organisms. It occurs in yeasts, both Saccharomyces and Candida. It occurs in plant parasitic nematodes, the ones that have that horizontal transfer from bacteria and fungi. It occurs in heliconius, but also in swallowtail butterflies. It occurs in East African cichlid fishes, which are rapidly evolving multiple phenotype change organisms, which have been something of a mystery. It's been observed in real time in Darwin's finches in the Galapagos. It occurs in sparrows. It occurs in dolphins. It occurs in flowering plants, sunflowers, orchids, and irises. And most interesting of all, it occurs in many crop plants, in emmer wheat, which is a primitive wheat, in flour wheat, which is what we use. Emmer wheat is a 2-species hybrid. Flour wheat is a 3-species hybrid. Rapeseed, tobacco, peas, coffee, potatoes, cotton, rice, soybeans, and quinoa. And so it's not as exceptional as it seems. And there's lots of evidence for what's called introgression in species, where they have sequences which come from a related strain, a related species, which are different from their own standard sequence for that function. So there's a lot of evidence of cross-species hybridization, and it can create a species quite rapidly. So it may be hybridization rather than selection, which is the engine of new species formation. Now, I want to talk a little bit about the role of biocognition in evolutionary change, because now that we know the evolutionary change is carried out by biological functions, cell biology and molecular genetics, we can ask, what's the role of the cognitive processes that regulate these systems in carrying out evolutionary change? And so here is some systems which are involved in biocognition initiating evolutionary change. So for example, quorum sensing, which is a process of pheromones, which allow bacteria to decide how dense their population is, can trigger DNA uptake and direct cell-to-cell DNA transfer in bacteria, which is enhanced in biofilms, which is where most bacteria actually live. on surfaces. We know in yeast, mating pheromones trigger oriented growth and cell fusions in yeast. We see this when yeast change mating type, and the A and the alpha cells grow towards each other and fuse to make a diploid cell, which is much more stable than the haploid cells. Abiotic stresses activate TEs, transposable elements, in all types of cells: wounding, heat, radiation, peroxide or reactive oxygens, species, toxic metals, insecticides, and other synthetic organic chemicals in one or another organism. These have been documented to induce transposable element movement through the genome. Biotic stresses activate recombinases, targeted DNA cleavages, transposable elements, and changes in ploidy in many types of cells. These biotic stresses can be virus infections, toxins, DNA transfection, predators, replicative stress, telomere erosion, inflammation, tissue culture, and the breakage fusion bridge cycle discovered by Barbara McClintock, where chromosomes fuse and break at every mitotic division. That induces a huge amount of instability in the genome. Starvation activates transposons and retrotransposons in bacteria, yeast, and mice. Interspecific hybridization disrupts epigenetic regulation, activates transposable elements, stimulates chromosome rearrangements, and can initiate whole genome duplications and rapid speciation in eukaryotes, ranging from yeast to flowering plants, insects, and vertebrates. So it seems to be a pretty widespread phenomenon. So the ability of stresses and different kinds of treatments to trigger some of these biochemical activities is an important role. And that's part of biocognition. There are also roles for biocognition in executing evolutionary change. So they coordinate cell engulfments during symbiogenetic genetic cell fusions, like the one that created the ancestor of all eukaryotes, fertilizations, as goes on every day among sexually reproducing organisms, and in pathogenesis too, where bacteria infect cells, for example, or other pathogens infect cells. They don't do it randomly. It's a coordinated process, and it needs to be controlled by some kind of feedback process. The establishment of mating pairs during horizontal DNA transfer in prokaryotes, that's making pairs of cells which can transfer DNA from cell to cell, and sexual reproduction in eukaryotes, that's having sperm and ovum unite, which is not a random process by any means. It's also involved in forming multimolecular nucleoprotein complexes needed for genome restructuring, such as DNA transposition, retrovirus retrotransposition, chromothripsis, and chromoplexy. Those are non-random events which need to be organized spatially with respect to the genome. There's the formation of intranuclear repair centers and recruitment of damaged chromosomes to them to generate rearrangements by double-strand break repair systems. So you irradiate a yeast cell, it forms a repair center, and multiple chromosomes with damage migrate to that center where repair and can take place. Just recognizing lesions in DNA damaged by the cell complexes, which will correct them, is a biocognitive process. And the response to altered epigenetic status in interspecific hybrids is also a non-random response. So it brings us to the question, which was forbidden to ask as long as people were sure there was no biology to evolutionary transformations, as long as it was just random change and natural selection. But could cells have the tools to guide evolutionary change adaptively in response to cognitive inputs? And here are some suggestions for where it may happen, and some of the evidence that would be relevant to answering such a question. First of all, cells might be able to interpret the nature of the trigger for genome change. So stress activation and movement by transposable elements and other mobile DNAs lead to cognate stress-inducible loci at the sites of insertion. So heat stress in maize activates the heat-triggered retrotransposon to insert, and the components of the maize heat stress response system have received their control signals from those mobile elements. There's the micronucleus trigger for chromosynthesis, and that's the source of complex genome novelty in a restricted region of the genome. Mobile DNA elements themselves are subject to several forms of targeting within the genome. So proteins can direct transposable element insertions to specific sites or to DNA in a particular functional state. For example, transcribed versus non-transcribed, or silenced. Some elements go preferentially to transcribed regions, and some go preferentially to non-transcribed regions. There are complementary insertion preferences for particular transcriptional and epigenetic states across the genome, and transcription factors are known to guide TE integration. In the early studies on LTR retrotransposons in yeast, they were seen to insert almost exclusively into tRNA regions, and it was later figured out that the integrase protein of that retrotransposon bound to a transcription factor for RNA polymerase III, which transcribes the tRNAs, and that was the source of guidance. And finally, complex phenotypic novelties frequently result from the formation of genomic islands. These are regions which are dense complexes of functions which are involved in encoding the ability to produce novel phenotypes in a specific in a strain which has them. And these genomic islands hold genetic clusters which have been associated with speciation-related adaptation changes in eukaryotes and horizontally transferred mobile adaptivity determinants in prokaryotes. And there are well-studied systems on bacterial plasmids which acquire cassettes of antibiotic resistance determining elements and other sequences, which are just the protein encoding sequences. And the integron provides a promoter for them, and they're expressed, and they confer multiple antibiotic resistance to superbugs. So I hope I've convinced you that there's something to the idea that change is biological and it's not random or physical in nature, and that we can make more sense of it by studying its biological properties. So thank you very much.
Thank you, Dr. Shapiro, for your very detailed, expert presentation. If
I
we
hope I wasn't too expert.
no. Well, I think it's given us a lot to talk about because we have some very we have some very wonderful scientists in the audience today, so they'll perhaps be able to feed up much more off than I know I can. So please, the floor is open for any comments or questions. And also, Dr. Shapiro, if you can just stop sharing your screen.
Oh,
Here, I think I can do it. Yes. Okay.
okay.
Yes, Dr. Roslenbridge, please.
Yeah. I have a direct question to Dr. Shapiro. Do you think there will be situations where such genetic changes can be generated by organisms in a directed way? So we just don't know this exactly right now. You made it somehow plausible, but do you think it will turn out that organisms are able to change the genome in a directive way?
Well, I think we have to entertain the possibility, and we have to go about trying to find the evidence for it. I think experimentally that can be done by taking interspecific hybrids, for example, and subjecting them to different inputs and asking whether that changes the changes that occur, whether there's any reliability in those changes. Nowadays, with the ease of genome sequencing, we should be able to do that with experimental organisms pretty readily.
Yeah, thank you.
Please.
Okay, yeah, if I might ask just a more general question. Thank you for this wonderful presentation, first of all. Just wondering, like, when you have overwhelming evidence of these kind of processes, and it's been established for decades, how is it that someone like, let's say, Richard Dawkins can still promulgate this kind of very narrow gene's eye view of evolution? And like, he recently published a new book about this, where he, of course, makes probably some gestures to new research, but still is largely, I don't know if it's willful ignorance or if it's just when you built your career on a different kind of view that you wouldn't like be willing to admit that there is like this there is something fundamentally wrong with this view. I don't know if you have any views on that.
Well, I agree with your suggestion at the end. And we have to remember that Richard Dawkins has made a lot of money selling books with his own version of things. And for him to go back on that, he may feel that threatens his future earnings. He held a debate with my colleague Dennis Noble at Heian Way in Britain a couple of years ago. And Dennis pointed out to him some of the things I showed, like the figures from the original draft human genome sequencing papers. And it hasn't changed Dawkins' mind. He's managed to rationalize to himself that this doesn't matter. We wrote a paper about this a couple of years ago called "What Keeps Modern or Evolutionary Genetic Biologists from Telling the Full Truth About Evolution." And they don't talk about it. Barbara McClintock doesn't appear in the evolution textbooks. That's an amazing result. it means they're not interested in the science. Barbara McClintock, her work has been known for decades. She got a Nobel Prize in 1983. That's 40 years ago,
Yeah.
41 years ago. So I think that there's been a circle of the wagons around the established paradigm and not taken into consideration any contrary evidence. And they cease to be scientists. They've become priests, I guess. I don't know what to say.
Yeah, if I may respond, I mean, I think, yeah, I agree with you, but I think also, at least the view that someone like Giuseppe Longo, for instance, he would say that, like you say, that they become priests. I guess another way of putting it is that they confuse. What has happened is that they confuse metaphors for theories, and
Yeah.
so like, and this is maybe like another question is like, I guess what we need, you could say, is like we need other metaphors, but we need metaphors that become theories instead of remaining metaphors or being confused or being theories. Like, I mean, metaphors like program, blueprint, these kind of things, you know, that is basically just taken from information theory and stuff like that, and just like transposed, you might say, like into biology without any kind of, you know, yeah.
Yeah, well, that's fine as long as you recognize that the cognitive capabilities of cells and organisms are included, because that's clearly at work. It just couldn't come out right otherwise.
Hmm.
You can't build a repair center and transport a damaged chromosome to it without knowing where it is and where the damage is.
Hmm.
And it's clear that these properties and capabilities are present in all kinds of cells. So I think it means a deep rethinking of what life is all about.
Hmm.
But it has to be based on science and scientific observation. But I think it's possible to, once you admit that possibility, and you can then think of the experiments in a pretty straightforward way. And we have the tools to do them. And if it's wrong, it will be shown to be wrong. But if it's right, it'll be further strengthened. And it'll be harder and harder for people to pretend that it's not right. I think also it's important to prepare a kind of agenda for the general populace about where evolution science is going. Because what's taught in the schools isn't going to change until there's very widespread acceptance among the general population.
Okay, thank you.
And do you think this process is on its way, the process of changing minds, or is there a lot of resistance within the established scientific community?
Yes and yes. It's on its way. There is a small group of us who call ourselves the third way of evolution. Neither Darwinism or intelligent design. And we are arguing for a much more open way of looking at the evolutionary process. And I think as people are beginning to wake up to the fact that biology is not just a matter of chemistry and physics. It's something else. There's more there. Philip Ball recently wrote a book about it called, I forget what the title was now, something like The Real Science of Life or The Real Science of Biology. And pointing out that biologists think differently now than they did 10, 20, 30 years ago. and there is a lot of resistance to it. But I think things will change, and the younger scientists and younger academics will become accustomed to dealing with these new phenomena and these new processes, and we'll be seeking answers about them.
If there are no other questions for Dr. Shapiro, we can transition to the interdisciplinary dialogue. Before doing that, I just wanted to share one infographic that Dr. Shapiro and Dr. Noble put together. Dr. Halkadale raised the question of why is it so difficult for working scientists to accept these empirical conclusions. The other side is how to educate the next generation of scientists properly, and they made this very wonderful infographic of the textbooks, what's going wrong with the textbooks. So I'm just putting this on the video for whoever sees it, watches this video, they can find this. Here's the link. It's super wonderful. Shows what's going wrong with the top, 5 or 6 textbooks, and they give all the details of symbiogenesis, to the transposition, epigenetics, everything, all these new findings. So please feel free to look at this if you're interested in learning more about it and learning how to educate the next generation of scientists about it.